Rarely has there been a new technology which has generated more acrimonious disagreement than genetic modification.
I've heard some people suggest that climate change has been due to GM crops, that the rising cancer rates in the community are due to GM claims and suggestions that have no relationship with reality at all.
There seems to be a lot of confusion when it comes to genetic modification. A little known fact is that virtually everything we eat has been genetically modified in some way, even organic food. So what's all the fuss about? Well, apparently it's all in the definition. Professor Peter Langridge was an advisor to the Legislative Committee that defined genetically modified food.
Well, it is actually quite difficult to define, and that causes a confusion. But generally, we talk about genetically modified food as food that's derived from organisms that have been genetically engineered.
So genetic engineering is one way of modifying the genetic material in food. Are there other ways?
Yes, there are many methods, and we've been using them for hundreds of years to try and improve the crop plants and the food that we eat.
But they're not as controversial as genetic engineering?
No, I think they're largely accepted. They've been used for a very long period of time. They've given us the diversity and the quality of food that we now enjoy. But genetic engineering has been the technology that people are very worried about.
Modifying the genetic material in food is not new. For centuries, plant breeders have been introducing new genes into their crops by crossbreeding to create hybrids.
In order to make a cross between two different plants, what we first need to do is get the flower before the pollen is mature. That's removed. In other words, the flower is emasculated. And we go and we get some fresh pollen from another source, like here, see? And we take these anthers and we drop them into the flower, and that will then affect the cross-pollination.
The variety in shape and colour of tomatoes we see today are all a result of crossbreeding. In the early 1900s, plant breeders began to get more imaginative with their techniques. They started crossing species that would never normally cross, like a wild wheat with a modern variety.
It's a little bit like crossing a Chihuahua with a Great Dane. It's possible, but it creates pretty serious problems for the organisms involved. And it's the same thing with wheat. So that even though you often will get fertilisation occurring, the seeds may not be normal. So what we need to do is rescue the embryo. And so there's our seed. And the embryo sits at the top here. And to get the embryo out, we cut in at the top. There's the embryo.
Right.
Okay, so we take that embryo and we can put it into culture, onto medium, and it will grow and develop into a normal plant.
Because the plant wouldn't grow normally without help, the extra nourishment is provided by these cultures. And this technique is the basis of all our modern wheat varieties. These techniques bring about genetic diversity, which is important for the survival and adaptability of a species. So by changing the combination of genes in a plant, you can make them more resilient to environmental stresses. Without genetic diversity, we'd be vulnerable to mass crop extinction. Originally from South America, the potato was introduced to Europe in the 16th century. It became the staple food in Ireland until the disaster of the Great Potato Famine.
Only a few varieties have been grown in Ireland. At some point, a disease got across. The disease was called late blight and virtually wiped out the potato crop. The result was massive famine in Ireland, with hundreds of thousands of people dying and many more being forced to migrate.
In the 1950s, plant breeders used chemicals or ionising radiation to deliberately cause mutations in the DNA of seeds. Once planted, they looked for new varieties. Today, this type of mutational breeding is an unregulated way of modifying the genetic material in food. Seedless mandarins resulted from this type of breeding. You've just seen ways in which the food you eat today, including organic food, can be genetically modified by techniques used for hundreds of years. What I'm about to show you is how to genetically modify a plant using genetic engineering, and it's this technique that the anti-GM movement wants stopped. Scientists isolate a gene or even engineer one in the lab.
The technology would certainly allow us to take genes from any source and put them into plants. But most of the work that we do is taking genes that are already in the plant and modifying their expression, or alternatively, genes from closely related species.
The new gene, attached to a circular piece of DNA called a plasmid, is inserted into a bacterium called agrobacterium, which naturally infects plants. Once its harmful properties are disarmed, the agrobacterium smuggles the new gene into the host plant.
What I've got is some Arabidopsis plants here, and these have got immature flowers, and all I'm going to do is invert the plant into the solution at about 10 seconds or so, and that is essentially it. There'll be a transfer of DNA from the agrobacterium into the female germline of the plant, and the DNA will be transferred.
So why don't we just stick to conventional methods for modifying DNA? Why do we need genetic engineering?
Well, the advantage of this technology is that you're able to transfer a single gene rather than a whole lot of other genes that come through when you do a conventional cross.
Rules regulating this type of technology began in the mid-70s and gradually became more stringent.
Most recently, in 2001, the federal government brought in the Gene Technology Act, which went from an advisory system to a regulatory system where there were penalties that could be imposed if someone disobeyed the rules and guidelines.
But it's these laws that some scientists say makes this new technology safe.
If I produce a product today through conventional breeding, there's no requirement for me to have any food testing done or human trials or anything like that. If I use an alternate technology such as genetic engineering, then I'm required to prove that that food is safe.
Our brief is to try and develop and deliver the best technologies possible to the Australian community. But we are concerned, I think, that there's now mistrust for scientists and decisions that scientists are making.